Hi, my name is Alea, and in this video, we're going to cover the prokaryotic cell envelope. So we've kind of already in other videos talked about what prokaryotes are and some of the things that are found inside of a prokaryotic cell. So now in this video, we're going to be covering the things that surround the bacteria. So we call this the cell envelope. So it's all of the layers, outside of the bacteria. So here's kind of that picture that we've been using for the other videos on a prokaryotic cell. Remember that a prokaryotic cell has a membrane, it's got DNA, there's no nucleus, there's some ribosomes, and there's some plasmids, right? And so now we're going to focus on all of this extra stuff. I haven't discussed it before, but bacteria and other microbes, they have a bunch of other layers to protect them, which makes a lot of sense. Remember, they are single-celled organisms, so they're just all on their own. And so obviously they need a lot of protection from the environment. So we've discussed the cell membrane in different videos, but just as a recap, the prokaryotic cell membrane is going to be made up of phospholipids. So it is a phospholipid bilayer. And then there are a bunch of proteins that are embedded in this phospholipid bilayer for communication, to adhere to the environment, whatever it is, transport. So there's a lot of things going on in the cell membrane. And as a reminder, remember that the archaeal cell membrane is slightly different. The phospholipids are arranged a little bit differently. So yeah. So now we're going to talk about the bacterial cell wall. The bacterial cell wall is this layer outside of the cell membrane that gives bacteria their structure, and it really helps them with osmotic pressure. So whenever a bacteria, for example, is in a hypotonic environment, as you probably know, in a hypotonic environment, water will enter the cell and the cell can expand and burst. But of course, that would be horrible for the bacteria. And so they have this cell wall. It's kind of like chainmail. It's really rigid and it helps give it its structure and also prevents it from essentially exploding. So the cell wall is really important for structure, and it is made up of this substance called peptidoglycan. Now, it is called peptidoglycan because it is a mixture of sugars and amino acids. So peptido, that root word, has to do with amino acids, and then glycan has to do with sugar. And so if we combine those two, that's what it is. So when we are looking at the structure of this bacterial cell wall called peptidoglycan, it is made up of two types of sugar, NAM and NAG. That is the shorthand name, by the way. There's like a super long name. You don't really need to know it, but NAG and NAM, those are the two sugars, and they alternate. So it's like NAM, NAG, etc. And they are linked to each other. And then there are also peptides. So basically a chain of amino acids, and those are the smaller balls that are shown in this diagram. And that is essentially what peptidoglycan is. Again, it is this layer to give bacteria their structure. Now, to give you perspective on where this is found, if this is this is the surface of the bacteria and this is their cell membrane, plasma membrane, the cell wall is typically right outside of that. So the layer above it. So if you think of the plasma membrane as the skin of the bacteria, then this cell wall would be like the clothes, if we were to use an analogy. So your skin would be your cell membrane, and then if you were wearing clothes, that would be your cell wall. And so yeah, that's what the peptidoglycan looks like. Now, in gram positives, they have a slightly different structure for the cell wall. By the way, I should have mentioned that there are two types of cell walls, gram positive and gram negative. So we group bacteria based on how their cell wall looks. And there's the gram positive bacteria, and then there's the gram negative. We'll talk more about the gram negative in the next slide. But yeah, the gram positives have a more, I guess, involved cell wall. So if you look at their cell wall here, this is gram positive. They've got peptides that, form these links, and then they also have peptides forming cross bridges. Whereas in the gram negatives, there are no peptides forming the cross bridge. Gram positives also have this teichoic acid that basically goes through their entire cell wall to give it even more structure. So that's shown here. So this is the entire cell wall of a gram positive, all in this light blue. And there are these teichoic acids that anchor it basically. And as you can probably tell, the cell wall for gram positives is extremely thick, The gram-negative cell wall is just this little tiny blue layer here. So gram-negatives have a very thin cell wall made of peptidoglycan, and gram-positives have a very thick peptidoglycan cell wall. So those are the two main differences between these two. And hopefully you can tell in this picture that the gram-negatives also have a bunch of extra stuff, which we're going to talk about now. But yeah, this is the cell wall for bacteria. And as I already mentioned, gram-negatives have basically an extra layer. So they have their membrane, they have a very thin cell wall made of peptidoglycan, and then outside of that, they have a second membrane. So they just have two membranes. They have two skins. They have their regular skin, and then they have clothes, and then they have more skin on top of that. And so gram-negatives have this thin cell wall, but they have this extra membrane, which we call the outer membrane. So usually we call the first membrane the inner membrane, and then the second membrane the outer membrane for gram-negatives. Now, this outer membrane is not the same as the inner membrane. So it's not just phospholipids. There are phospholipids, but it also has all of these special things. So one of the main things that is found in the outer membrane is this lipopolysaccharide, which is shown here. And LPS is the short name for it. It is just a different type of lipid. So as you can see, there's these fatty acids, but then there's like this entire structure that's attached to it. And so it's got this lipid A component, and then it has this like core made of like sugars and stuff, and then there's this O antigen that is attached to it. So it is a relatively large molecule compared to just a regular phospholipid. Now, why am I mentioning this? Because later when we talk about infections of bacteria, this O antigen and lipid A are really important for that whole like infectious stage of gram-negatives. Okay, there is another special type of bacteria that doesn't really fall under, I mean, I guess it's technically a gram-positive, but it's like a subcategory of the gram-positives. They are called the acid-fast bacteria. So they are still gram-positive. So as you can see, they've got one plasma membrane or cell membrane, and then they have a very thick peptidoglycan cell wall. But outside of that, they just have these mycolic acids. It's not necessarily a second membrane. It's just these molecules that are attached outside of the cell wall. And there's really only a few bacteria that are acid fast, and they're all, well, primarily going to be in the Mycobacterium group, which includes Mycobacterium tuberculosis, which causes tuberculosis. So in terms of, clinical settings, they are really important. So yeah, acid fasts are a subcategory of gram-positives. They basically look the same except for this additional mycolic acid molecule that they have. All right. And so those are the two main groups. So we've got the gram-positives and the gram-negatives, and that's what they're, what these like extra layers that they have. But guess what? Some bacteria have even more bonus protection. And I'm not only going to really talk about a few extra things. So one is capsules. And by the way, this is, as I mentioned, this is bonus, meaning that not all bacteria produce this. And actually, even in a specific type of bacteria, sometimes they'll produce it, sometimes they won't, just depending on their environment or how they're feeling. If they're feeling icky, maybe they'll add extra layers, right? It's kind of like us, like, oh, it's cold outside, I'm going to add like an extra coat. It's just whatever they're feeling that day. They're just vibing. Anyway, so some bacteria can produce capsules. Capsules are made of sugars and proteins, or both, or one, whatever. And they just kind of like secrete this stuff out. And it adds this extra layer of protection. This is what capsule looks like, by the way. So here are the bacteria, and the white layer around them is all of this like capsule that they're secreting. Some bacteria produce slime layer. What is the difference between a slime layer and a capsule? It's just how tight the molecules are. Capsules have really tight sugars and proteins, whereas a slime layer is more loose. But it's basically the same thing, just extra stuff that they're secreting for protection. And then the S layer. Now this I hate. Why? Why is there slime layer, S layer? It's the same, like it almost sounds the same, but the S layer is a specific type of layer that's produced. It is a lattice structure, so it's kind of like a mesh, and is made up of proteins or glycoproteins. But again, same thing, it's just extra layers. And this one is anchored to the cell wall. So the slime layer is not anchored to the bacteria. You can actually like it can actually come off, whereas the S layer is like anchored to the bacteria. But either way, all of these are just extra bonus layers that the bacteria can have. Again, some bacteria have it, some bacteria don't. It's just whatever, however they're feeling. So, and also whether or not they can make it. So yeah, those are just the extra things that some bacteria have. Hopefully this was a fun video. I will see you in the next video.